Hello guys, welcome to my YouTube channel. So in this video, we are going to talk about glycogenesis. We are going to generate glycogen. So if you read it backwards, genesis is the generation, or we are going to generate glycogen. Now, when does this process occur in our body? Where does it occur and why does it occur? And what are some of those the enzymes or the organs involved in glycogenesis? So all that we are going to answer in this video. Now, glycogenesis is a process that occurs when there is excess glucose in your body. So when there is excess glucose in the circulation, the body will decide to preserve some of the glucose into a form that can be used later, or into a way that you can get that glucose later. Because actually there are certain cells in the body that entirely depend on glucose. For example, like the brain cells and sometimes the erythrocytes. These cells entirely depend on glucose as the source of energy. So we need to make sure that there is adequate glucose always, because remember, our brain cells need to keep on working. They must never stop. So remember, we must make sure that there is always glucose. So when there is excess glucose, we can find a way of conserving some in a form that can be accessed later. Now, let's start. When this is going to represent the cell, then this space here is going to represent the circulation. So this is our cell. So when there is excess glucose in the circulation, the glucose is going to get to the cell by the blood receptors. So I'm going to say this is a liver cell. So when there is a liver cell, the glucose molecule always has 6 carbons. So this is our glucose. Glucose molecule is going to get into the cell by blood receptors. So in the case of glucose, in the case of liver, we are going to have blood 2 receptors. Blood 2 receptors. These receptors are going to allow the passage of glucose into the cell. So when glucose gets into the cell, so we have our glucose molecule here. 2, 3, 4, 5, 6. It's going to be acted upon by an enzyme. So what is the name of this enzyme? This enzyme is going to be called glucokinase. It is a kinase. It means it's going to phosphorylate. It's going to add a phosphate group. So where are we going to get that phosphate group from? We're going to get it from ATP. So that's going to be reduced to ADP. It's going to rip off that phosphate group from ADP. Then it's going to add it to glucose. So I'm going to have a 3, 4, 5, 6. Then it's going to add a phosphate group at position 6. What's the name of this one? This is called glucose 6-phosphate. Glucose 6-phosphate. Now, some of you might think that you are going through glycolysis. You want to see what's going to happen. Now, glucose 6-phosphate, instead of going through glycolysis so that it will be converted to fructose 6-phosphate, we are going to convert this glucose 6-phosphate into glucose 1-phosphate. So 1, 2, 3, 4, 5, 6. So we are going to bring this phosphate group from position 6 to position 1. What's the name of that enzyme? This enzyme is called phosphoglucose mutase. It's going to convert glucose 6-phosphate into glucose 1-phosphate. Glucose 1-phosphate. Alright. To me, it's just going to take the phosphate group from position 6 to position 1 so that we have glucose 1-phosphate. Alright. Now, inside the cytoplasm, we have something called UTP. This is called uridine triphosphate. Uridine triphosphate. So uridine triphosphate is going to react with glucose 1-phosphate. So see these guys. They are going to react in the presence of an enzyme. This enzyme is going to be called UDP glucose pyrophosphorylase. Pyrophosphorylase. Alright. What this enzyme is going to do, so this is uridine triphosphate. So it's going to form pyrophosphate, which has two phosphate molecules. Alright, and also it's going to take this phosphate group here. So remember, this is going to be cleaved. So we're going to take, so it's UTP glucose. So it has three phosphate molecules. We're going to take out two. So you're going to have UDP, right? I mean UMP, because you did monophosphate. It's going to take this phosphate group from glucose 1 phosphate. So that will form UDP. So it's going to form UDP glucose. It's going to also going to take the glucose molecule. So it's going to form UDP glucose plus pyrophosphate by the help of UDP glucose pyrophosphorylase. Now guys, let's see. Inside the circulation, there's also a protein molecule. This molecule is going to be called glycogenesis. It has a whole H group. This is called glycogenesis. Alright. This pyrophosphate, the two phosphate molecules, can be acted upon by an enzyme. Now, let me draw them this way. Can be acted upon by an enzyme so as to form individual phosphate groups. What's the name of this enzyme? This enzyme is called pyrophosphatase. Alright. Pyrophosphatase is going to break the bond between the individual phosphate group between the phosphate group to form individual inorganic phosphates. So in the process, there is going to be generation of some energy. So this is the energy. What is this energy going to do? This energy is going to be used in a process called autoglycosylation. Now, let's have our UDP glucose here. So these are UDP glucose. Alright. Now, this UDP glucose, through a process called autoglycosylation, is going to be added onto glycogenesis with the help of this energy here, generated from pyrophosphatase acting on or breaking down the pyrophosphate into individual inorganic phosphates. So when you are forming a bond, this edge is going to be removed. Alright, so I'm going to form a bond there. Alright, so I'm going to add glucose, 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 glucose. But whenever we add this UDP glucose, we're going to get rid of those UDP. So you just use this circle to represent the glucose. So you can just say it's looking like that. The UDP right to glucose. But remember, the bond between individual glucose molecules is called alpha 1,4 glycosidic bond. Alpha 1,4 glycosidic bond. This bond here. It can also form alpha 1,6 glycosidic bond, but you are going to see. So the bond between this glucose and this glucose and this glucose is called alpha 1,4 glycosidic bond. So this process is called autoglycosylation. We are adding them without involvement of any enzyme, right? So it's called autoglycosylation. Right. Now, but remember, this energy should be depleted. You do not add enough of these glucose molecules to form glycogen. So there's another enzyme that is going to come into play. This enzyme is going to be called now this enzyme is going to be called branching enzyme. Branching enzyme. So let me just drop this here. Alright. What is this branching enzyme is going to do? But before we get to branching enzyme, there's another enzyme that's going to come before branching enzyme, so that we can make sure that we add enough of these glucose molecules to form something that's very heavy. So what is that enzyme? That enzyme is going to be called glycogen synthase. Alright. You have glycogen synthase and branching enzyme. They are going to come into play. So we're going to have glycogen synthesis first. So what is glycogen synthesis going to do? Glycogen synthesis is going to take these UDP glucose molecules, add them on top, because we have depleted the energy that we use for autoglycosylation. So we're going to have glycogen synthesis. So it's going to add more of these UDP glucose molecules. So we're going to add more and more and more. Alright. So whenever we add a glucose molecule, we're going to get rid of UDP. So whenever we form alpha 1 for glycosidic bond, we're going to get rid of UDP. Alright. Now, this chain is getting too long, so you must find a way of, you see, manipulating and fixing it inside so that you can have a small, you can take a small space. So you just want to fold it so that you can take a small space, right? So what are we going to do? Is when this enzyme is going to come into play, the branching enzyme. It's going to break the alpha 1 form glycosidic bond and form alpha 1 6 glycosidic bond. So, see here. This chain is going to go too long so that you are not going to package enough glucose molecules. So, it's going to come here and break some of these. Maybe until then. Then, so let's see, this is the 6th carbon in the glucose molecule. So, it's going to form alpha 1 6 glycosidic bond and fix them back. Alright. This is going to create a room for the glycogen synthesis. Then it comes again. Add more of these. Alright. The same thing again happens. Branching enzyme comes again. Breaks some of these. It can make either here. 1, 2, 3. All those glucose molecules. So this process is going to go on like this. So we're just going to form more of these glucose molecules are going to be packaged, so that we have a very big and folded structure. So what do you call this? This is what we call glycogen. So a lot of this is called glycogen, and that is the aim of the process here. Glycogenesis is just the way of packaging enough glucose molecules into glycogen so that glycogen can be stored in the liver for later use. So we have the glucose molecules. Alpha 1 4 glycosidic bond, alpha 1 6 glycosidic bond, the enzymes, branching enzyme, glycogen synthesis, so that we finally have a glycogen molecule. So glycogen molecule, when there is sometimes need, so when we need glucose in our body, we can now break down the glycogen molecule in a process called glycogenolysis, which I'm going to discuss in our next video, so that we get the glucose molecule so that they will be taken where they are needed. So I say glucose molecule will get to the cell via GLUT receptors. Now, it's going to be phosphorylated by an enzyme called glycogenesis, which primarily exists in the liver, to glucose 6-phosphate, which is again going to be acted upon by an enzyme called phosphoglucosamidase to glucose 1-phosphate. Glucose 1-phosphate is going to be react with the uridine triphosphate in the presence of UDP glucose pyrophosphorylase to give us UDP glucose and pyrophosphate. Now, pyrophosphate is going to be broken down by pyrophosphatase into individual inorganic phosphates, leading to generation of some energy. This energy is going to be used in autoglycosylation, where we are going to take these UDP glucose molecules, add them onto glycogen to generate a very long chain. Then, to generate a chain, but because it can't be long enough, because this energy is going to be depleted, glycogen synthesis is going to come into play, add more until you get a very long chain. Then a branching enzyme comes into play. It's going to form alpha 1 6 glycosidic bond to package enough of these glucose molecules into glycogen for later use. So I'm going to share with you the disorders that are related to glycogen storage or glycogen. So let me use this part. Now we have a glycogen disorder. So that's called glycogen storage diseases. Glycogen storage diseases. Alright. The first one is going to be called compressed disease. Correct. Pompe's disease is a disease that occurs when there is a deficiency of an enzyme called lysosomal maltase. Lysosomal maltase is an enzyme that breaks this alpha 1 6 glycosidic bond so that you can have the individual glucose molecules, right? So when you don't have lysosomal maltase, there will be no way you can break down this individual these alpha 1 6 1 4 glycosidic bond mostly in the lysosomes, right? So we want to have enough of this glycogen being stored, but you can't break it down. So when you can't break it down, this is going to affect the cell's motility. The cells cannot easily move around, right? So when there's inadequacy of cell motility, this is most significantly seen in the heart, so that the heart cannot contract, right? So when the heart cannot contract, it can lead to heart attack. Heart attack can lead to death. So Pompe's disease mostly occurs in the heart. Now, there's less motility. Less cell motility. Cell motility. Leading to heart attack, then can lead to death. Number 2, not to have McArd's disease. This disease mostly occurs in the muscles. When there is lack of an enzyme that you're going to see in glycogenolysis, and you breaking down this glycogen molecule called phosphorylase, glycogen phosphorylase, which is also going to break the alpha 1 4 glycosidic bond. So when that enzyme is not there, then you cannot break down this glycogen molecule. So there will be accumulation of a lot of glycogen inside the muscles, right? So when the glycogen accumulates in the muscles, we're going to have problems like maybe during running, during exertion. We're going to have pain, you're going to feel pain in the muscle, and you're also going to feel exhaustion. So exhaustion mainly occurs when there's depletion of ATP and pain, lactic acid, but also there's relation to McArd's disease. That is when there's accumulation of glycogen inside the muscles, and this will lead to pain and exhaustion. Number 3, so this may occur in the muscles. Now, when there's lack of glycogen phosphorylase. Then number 3, we're going to have hypoglycemia. Hypoglycemia. Hypo means less, so there's less glucose. So this condition is mostly associated with less glucose or lack of glucose inside the body. So this mainly occurs when there is lack of an enzyme called glucose 6 phosphatase. So glucose phosphatase. So when this enzyme is not there, you cannot convert glucose 1 phosphate into or glucose 6 phosphate into glucose molecule. And mostly you are going to see this when breaking down this glycogen molecule into individual glucose molecules. So this is all about glycogenesis. We've converted or we've packaged glucose molecule into glycogen, which can now be broken down for later use when we now need glucose. So thank you so much, guys. If you found this video helpful, please subscribe to my YouTube channel, like and share. Then I'll appreciate that. Thank you so much, and make sure you keep in touch with me for more videos pertaining to metabolism and other processes that occur in the body.